Welcome everybody, and we're going to go through a really exciting subject today, deformulation of pharmaceutical RLDs. So for those of you who aren't aware, RLD stands for Reference Listed Drug, and we do a lot of this type of testing here at EAG. We are certainly the world leader in this type of testing, and I'm really excited to go over what we do, how we do it, and why we do it. So a little bit about myself. I received my master's degree in chemistry from the University of Missouri St. Louis. I started at EAG, which back then was EAG. Now we are Eurofins EAG, about 6 years ago. And I worked for about 3 years in the laboratory doing a lot of different analytical testings, including chromatography and spectroscopy, as well as these more complex projects like these deformulation activities, which included method development, custom sample preparation, and going through these difficult excipients that are kind of similar to each other and figuring out how to get from point A to point B, which would be start to finish of a successful project. So in today's webinar, we're going to talk about several different topics, including Eurofins, an overview of the Q1, Q2 guidelines, some common characterization techniques that we have, the process and how it works when you want to do a project with EAG, some example case studies of a topical, which you can think of ointments, creams, a solid oral, think of tablets, pills, capsules, and ophthalmic, which would be things like eye drops. And then we'll summarize. So Eurofins EAG is a global company. We have laboratories all over the world. And since we've become part of Eurofins, we're even more a global company. Eurofins is one of the largest companies in the world when it comes to analytical, life sciences, and engineering sciences testing. So here you'll see our smart chart, and a lot of the techniques that are listed here aren't going to be as applicable to a deformulation, but we are the world leader in surface and advanced imaging characterization, as well as several other different techniques. So you'll see several of these techniques reviewed again later in the presentation, but just know EAG as a whole has a lot of different techniques and a lot of different capabilities beyond just reverse engineering of pharmaceuticals to help you if you ever need that help. And feel free to reach out anytime. So let's get into the subject we all want to talk about, which is this deformulation process. So why are we actually doing this? So right now, the main goal is these early stage competitive product analyses. A name brand product in today's age costs quite a bit more than a generic product. So the generic companies will approach us and say, hey, we want to deformulate this material. We want to know what's in it so we can meet the Q1, Q2 equivalency guidelines set out by the FDA. And thus, you save a lot of time and money as a company by doing this process rather than creating a whole new product. So it's really useful. It's great. It's a great way to get generics to the market quicker. And we love being a part of that process. So we can also do things like API polymorph determinations and other characterizations around the API. We can do great comparisons of complex excipients. So think of materials that are not discrete peaks, discrete analytes. So for example, propylene glycol is one discrete peak. But if you have something, let's say HPMC or hydroxypropylmethylcellulose, something like that is going to have a range of these different materials within it. It's not just one molecular weight size of this HPMC, it's going to have a range. And we need to look at different grades of HPMC to compare them to the RLD or the innovator's product. When we talk about impurity identifications, this can be a number of things. We've identified contamination on tablets. We've done impurity investigations when it comes to degradation studies of APIs. We can even look between a good lot that's performing normally and a bad lot to look why are they different. And then when we talk about these deformulations, we can actually look at the locations of excipients in capsules, in transdermal patches. We can look at different dosage forms to understand how they are different, so on and so forth. So there's a lot that goes into these Q1, Q2 projects, but there's more that we can do to help. So when we talk about what Q1 and Q2 are, Q1 means the sameness of components. Basically, you need to have the right grade of material in your product to match the innovator. How we do this is for non-discrete analytes, and when I say discrete, that means they are one thing. It's the same molecular weight for every molecule in the reference material that you have. Whereas we have some more complex excipients, like I talked about HPMC, where one standard to another with different amounts of hydroxyl groups or propyl groups can really make a big difference in the overall behavior of your formulation if you use the wrong grade. So it's really important to meet that Q1. We also talk about Q2, which is the sameness of concentration. So when we talk about Q2, everything comes down to this 5% limit. We need to be within 5% of the actual value in the innovator's product, in the RLD formulation. And that's really important. You're going to hear that a lot today, that 5% number is really important. That's what matters at the end of the day. And that's what we always try to hit with our studies. Now, the one thing to note about all reverse engineerings of Q1, Q2 FDA submissions, we treat every product as an unknown, and they are unknown. We don't know the concentration going in, and that 5% limit is something that we have to use a lot of our different testing tools to evaluate how we're doing. So we'll talk about some of our strategies to do that as we go through this presentation. One other note, when we talk about this Q1, Q2 reverse engineering project, we have designed our SOPs to test for these products. We are very well suited in doing it. And we can also adapt if there are other agency requirements. If you're submitting to a different government agency or a different agency altogether, we can certainly adapt our methodologies. So what have we worked on? And the answer is everything. We've seen it all. We've seen it all several times. So most of the time you'll see that we have topicals, solid orals, ophthalmics, transdermal patches are becoming more common. So we've seen a lot of different types of materials, and we've seen anything from something off the counter to drugs you probably haven't even heard of. So we have a lot of experience, and we're always more than willing to kind of walk through what we can do and how we can help. So like I said, we've seen it all. We've actually analyzed over 220 unique RLD products. Now, that number is a little bit misleading. We've actually analyzed over 500 different RLDs. Now, when I say different, I mean a different project. We've seen several of these RLDs several times. So with that said, most of what we see are topicals and solid orals, and then solution type materials as well. So we've seen a lot of these materials. We do these deformulations all the time. We almost always have one in-house, if not more. So we're always working on them, always developing methods, always getting better. So let's get into the process of an RLD deformulation. So where do we start? Well, we always start with a project manager, someone who's going to be able to direct the project within the lab and coordinate with our clients. We like to have weekly to bi-weekly updates through email, calls if needed. We can also source RLDs. So one thing that a lot of our clients like to do is send us RLDs. We can also procure them if needed. And that is something that we often offer to our customers. When it comes to the methodology, typically for chromatographic techniques, we're going to do duplicate sample preparations and matrix spikes. When it comes to non-chromatographic techniques, think of ICP, think of NMR. Typically, we're either going to do standard additions or we're going to run calibration curves alongside. Generally speaking, we'll do triplicate preparations when we can't do multiple injections from the same vial, which is true of most chromatographic techniques with an auto sampler. Reporting is going to be something that we are always very aware of. That's your, as a customer, that is your data that you need to submit. That's how you get to where you need to get to. And we're always aware of that. And we want to make sure we provide the information needed within the report to get you to those goals. Some methodologies may be proprietary though, and we can discuss that when we actually have a project going on with our clients and make sure they're aware of what we can and cannot share in that case. And then timing, right now, we are still looking somewhere in the 4 to 6 week window, 20 to 30 business days to complete projects like this. So we have a lot of experience doing this and we're getting better all the time and faster all the time. So what exactly needs to be done to make these projects successful? Well, Again, we hit on this fact, these are unknown materials. When we get the materials in our door, the RLDs, we don't know what's in them. So the first thing is we need to screen the materials. We need to get range finds. We need to figure out roughly how much of each ingredient is present so we can build calibration curves. We need to look at different complex excipients, different grades of those complex excipients, and compare them and identify the correct one for your formulation. Again, Q1 is important. We have to have the same grades. When it comes to how we proceed at that point, we generate these multi-point calibration curves where possible with authentic reference standards. Then we do duplicate preps, duplicate injections, as well as a matrix spike. And if you're unfamiliar with the matrix spike, what that is you take your quantitation. You have a known value of an ingredient in your RLD. Then you add a known amount of your reference material into that RLD. And from there, now you should have a known amount that you've quantified in the RLD. You've added a known amount, and we need to make sure we can recover all of it. We need to make sure that when we run it through this chromatographic system, that we can see everything we added in plus everything that should have been there. And the hope is that we get 100% recovery. And that's kind of how we look at a matrix spike. Now, obviously, a lot of it has to do with the method, with the matrix. So what are we looking for with the matrix spike? We're looking to see, is the matrix suppressing our response? Is it enhancing our response? Do we see a different shift in the peak? One thing a matrix spike can provide is specificity of if we add a known material in, do we see the peak grow or do we see a new peak show up? These are all really important things with a matrix spike. So one thing that we ask, especially when there's complex excipients, is if you can supply placebos or formulated batches, that's going to make our methodology so much better. And what I mean by that is if you provide us a formulation similar to the RLD or even different, but with the same ingredients, the matrices should be similar and we should be able to evaluate our method. If we know what's in your placebo and we're hitting those numbers, that means our method should be working really well. If we're not hitting those numbers, we know we need to adjust. If you send us a placebo or formulated batch and don't tell us what's in it, it's really just doing two deforms at that point. We're looking at the RLD, which is unknown, and your placebo, which is unknown. So that's why we asked that you would share that information if you wanted to move forward with the project to make this successful. Then we also look at, the report. Typically, we're going to send you a draft when it's complete. And then after you've had time to review, talk to our team, talk to your team, come back to us. We can send a final report once you're happy with it. And then again, back to the program director, weekly updates as to the status of your project and what is going on, because these are 4 to 6 week projects. We don't want to leave you in the dark for 6 weeks. So that's kind of an overview of what you get with EAG. Let's talk about the process. So initially, it comes into someone like myself, where you say, hey, I want to deformulate this material. So I'm going to go find the package insert. I'm going to find the different excipients that we might need. I'm going to look at the techniques that we might need based on the excipient list that we have. And I'm going to look at how long do I think it's going to take. I'm going to put that all together into a proposal, into this project that I've designed, and send it to you. And that would be our contract. And once you've agreed to move forward with that, we're going to have a project kickoff with that program director. Now, the interesting thing is the program director is going to do exactly what I did. They're going to go through, they're going to look at the insert, they're going to determine what grades of materials we need to get. They're going to look at the analytical techniques that we might need, and they're going to map it out in a different way than I did, by excipient rather than as a project as a whole. So that project kickoff happens. Then we go into that preliminary testing and screening, basically doing our range finding, doing all the testing that is required to identify the Q1 parts of the formulation and give us that range or that estimated concentration so we can set up the next step, which is those quantitations. So once we go through the quantitation steps, that's when you should be seeing periodic updates as we make progress. And finally, you'll get a report with the concentrations of the materials that we've identified and quantified in the formulation as requested. So what does an update look like? Well, in this case, an update is going to show, here are the excipients, here's the material we're using, here's the technique we're using, here's the target due date, and then the concentrations. So in this case, you'll see we have 3 lots. We want to see lot-to-lot variation. Typically, we want to know, the RSD of that study, how tight are our results. And then at the end of the day, we want to show you what we have done, what we expect to have done, give you any updates, anything that's challenging, so on and so forth. So an update is really important in this case because this is where we interact with our customers. This isn't not a process that is solely send us something, we send you something. This is an iterative process. We work with our customers to make these projects successful. And we always ask that when you send us materials, when we ask that if you have anything else to send us, just let us know. We want to make sure that at the end of the day, these projects are very successful. So reporting an RLD deformulation is also very important. And we can report it in several different ways. As you'll see on the left, we have average concentration by weight percent. And on the right, we have milligrams per tablet. So this is obviously a solid oral formulation. And we can report it in a number of ways, like shown here. So as long as our initial weight percent value that we calculate can be updated into these other formats, we can provide data in any way requested as long as we're not changing the data. So there's a couple things to note here. If you look at the very bottom under total recovery, you'll see that adds up to 99.95%. And you'll think, well, that's not 100. Well, actually, that's a really, really tight deformulation. The closer we get to 100, obviously, that's great. We want to be there. But with these deformulations and any scientific measurement, really, you have error associated with it, whether that be the balance, whether that be the instrument, whether that be the integrations of the different softwares. There's some error there. And as you see with the RSD and RPD section, these errors can be associated with these different percentages. This is basically our precision. And as you can see, what if one of these materials is 1% high and another's 1% low? Just think of it in that regard. Well, it still adds up to 99.95, but you're a little off. Well, that's why we have the matrix spike. That's why we have these other parameters to look at. We evaluate our data in many different ways. So typically what I would say for a successful deformulation, we're looking anywhere from 98% up to 102%. Of course, factoring in that error. But at the end of the day, the important thing is plus or minus 5% of the actual value. That's what we have to hit. In theory, everything can be 4% high, and we could be at 104%, and that's still successful. Now, that's not what we aim for, but that's just how you need to think about these deformulations when we get into it. Plus or minus 5% is always the goal. So now we've talked about the process, let's get into some of the excipients and what do we come across that is really challenging or difficult? Well, almost everything on this list has one thing in common. It's not just one thing. So when you think of mineral oil, it's not a single type of hydrocarbon. It's a lot of branched hydrocarbons all added together to give you a range. When you think of white petrolatum, we have these branched and we have straight chain hydrocarbons. So mineral oil and petrolatum are probably the most common complex excipients that we face, and they're almost always together. So they're really difficult together in the sense that you can't physically separate mineral oil and petrolatum. They are chemically very similar. There's no way to remove a straight chain from a branched hydrocarbon. They both have branched hydrocarbons, so there's no way to remove them from each other. So how do we tackle something like that? And we'll get to that in one of the later case studies, but I'll kind of cover it right now. Typically, what you're doing is you're isolating those materials from everything else. You're running it through a GC chromatogram. You're identifying the grades of mineral oil and petrolatum. And then from there, you basically need to either have placebos, have mixed standards of different materials to compare against. The process itself is not straightforward. Really, we're going to quantify total hydrocarbons, and then we have to speciate from there. How much of this total hydrocarbon is mineral oil, petrolatum? What if there's more hydrocarbons than just mineral oil and petrolatum? And we've seen that before. This can be really complex, but it's really exciting because we've developed a lot of different methodologies to be able to tackle these problems. These things that we've seen before, we've seen them a lot, and we've developed some really cool things to get us from point A to point B. Now, petrolatum, mineral oil, hydrocarbons are not the only difficult things that we see. Celluloses are some of the most difficult materials that we come across. And a lot of the reason for that is they're very similar, and a lot of the times they're not soluble like a petrolatum or a mineral oil. So MCC, microcrystalline cellulose, CMC, carboxymethyl cellulose, they don't go into solution very easily, if at all. HPMC, and ethyl cellulose can go into solution, but again, very difficult. They're not just one thing. They're a broad spectrum of things. Those things being celluloses, modified celluloses. And they're difficult. But we have come across ways, we have developed methods to approach these in a way that we feel really confident in giving you really good data when you need it. So the other things that we see as you go down this list, what they really have in common is they are not just one thing. They are broad ranges. They are difficult to solubilize. At times, they are difficult to remove from each other, and you might have to take on a xanthan gum and a lecithin at the same time, and you may not be able to separate them. So there's a lot of difficulties, and we've come up with a lot of different approaches to go about that. So not only do we do deformulations, we do method development and optimization. We do contaminant identification, like I talked about, spots on tablets. We can do impurity investigation, so think of degradation studies of APIs. We have a whole synthesis group to do custom synthesis. We can do imaging and mapping of excipients in tablets and other formulations. And we can also do particle size distribution of APIs and other materials if needed. So a lot of really cool things going on, and we're going to go through a couple of these right now. So when we talk about this imaging and mapping of APIs and excipients, there's several techniques we can do. So one of them is optical micrograph, like you see here. We have SEM to look at elemental profiling. We have TOF-SIMS, which we'll see in a minute, to look at materials. But at the end of the day, we want to be able to distinguish, hey, here's what's going on in the tablet, here's what's going on in the capsule, in the bead, that is something that's able to give our customers more data than they had before that helps them at the end of the day. So this is a really cool representation of the core and the coating that you can very clearly see, something that we've developed that's really cool to look at these different materials. So like I mentioned earlier, we can also do TOF-SIMS, things like SEM. As you can see here, we are looking at cellulose, sodium, HPMC, and succinate in the same material. And as you can see here, we're able to pretty clearly differentiate where are these materials within this tablet. So a really cool thing we're able to do. We do it quite often, and quite frankly, all we need is a standard of an appropriate molecular weight, because TOF-SIMS is obviously an MS technique, a mass technique, I should say. And then we need the RLD, and we can do this, and it works really, really well. So when it comes to the Raman particle sizing, so we've actually purchased and obtained several new instruments in the past couple of years through the support of Eurofins, and we're able to do some really cool stuff now that we weren't able to do in the past. One of those is this API particle sizing. So again, standards are very important. We need to run a standard. We're able to identify the API spectrum. And then we go through and we look at the tablet. We need to obviously have the rest of the spectrum, so things like celluloses, saccharides, lubricants, caking agents, things like that. And we're able to pull it out and provide you with something like a D10, D50, D90 setup of your particle sizing. So it's really cool to think that without actually having to go through a laser diffraction tool or another type of particle sizing, we're actually able to pull these particles out basically and give you a range of particle sizes, something that we're really proud of and that we do for several of our customers. And it's really, really interesting data, and it works very well. So we're really happy to offer this. So now that we've gone through the background of this testing, let's get into some individual case studies and how we approach those difficult problems. So the first case study I want to go through is an example of a topical ointment. So topical ointments tend to have hydrocarbons. Like we talked about, hydrocarbons can be really difficult. In this particular formulation, you're going to notice 3 hydrocarbons: petrolatum, mineral oil, and beeswax. So what makes this formulation so challenging is that when you think of these different materials, petrolatum, mineral oil, and parts of beeswax cannot be separated from each other. There's no physical way to take the waxy portion of beeswax and remove it from the petrolatum mineral oil. So how do we tackle that? Well, when it comes down to it, the first thing we're going to focus on is the easy stuff, or the quote unquote easy stuff. And that's going to be the propylene glycol, the phenoxyethanol, and the water. So these are all discrete analytes. You can order the same standard that you would order from one vendor versus another. We're going to quantify the propylene glycol and the phenoxyethanol, likely through GC, gas chromatography, and the water through Carl Fisher. So we have that done, that's out of the way. Now we're going to tackle the bigger problem. How do we do these hydrocarbons? Well, the first thing that we need to look at is identifying the grade of material in our RLD product. So when we talk about identifying grades, what we're going to do is we're going to screen against the RLD the different grades of petrolatum, mineral oil, and beeswax that we have available to us. Generally speaking, we like to screen at least 5 of each when available. Sometimes we can't get 5 standards. Sometimes we need to look at more. And what you notice is, and please follow my cursor now, what you'll notice is we have 2 different peaks kind of showing up here. We have these tall, more Gaussian peaks. Those are associated with the discrete hydrocarbons, the straight chain hydrocarbons of the petrolatum. Then you'll see this kind of rise in baseline as you follow my cursor. That is the mineral oil, and those are branched hydrocarbons that are not giving you a discrete peak, they're actually giving you a range, and that's where you get the actual rise in baseline because they come off in this time. They're just constantly flowing off because there's no separating them like the discrete or straight chain hydrocarbons. So when we actually get into tackling a problem like this, we've identified grades of the mineral oil and petrolatum. Then if you look at the right-hand side of our screen, one thing you're going to see is how do we tackle that beeswax? Because we know it adds a portion into our petrolatum and mineral oil. Well, the answer is beeswax actually has, and other hydrocarbon species can contain what we call marker responses, things that are unique to that material. And beeswax, fortunately for us, has these fatty acids. Think of stearic acid, linoleic, oleic acid, palmitic acid, these types of materials. So we were able to do what's called a FAME prep, fatty acid methyl esterification prep. We're able to visualize these peaks by gas chromatography. And once we've identified the correct grade of beeswax, which we can do based on these unique responses, we get the right ratios for that standard. We quantify using that standard. Now we have a percentage of the beeswax. So now we've done almost all the ingredients. All we're left with is mineral oil and petrolatum. Well, as you can see in the bottom left of the screen, the mineral oil and petrolatum is actually given as one concentration. Again, we can't separate them. So what do? Well, we've identified a grade. So now we're going to take an individual sample of mineral oil, petrolatum, beeswax, and our RLD, and we're going to run it through an extraction method that we developed internally. We're going to basically end up with a certain percentage of hydrocarbons left for each standard. For mineral oil and petrolatum, we would expect 100% recovery. For beeswax, likely we're going to expect somewhere in the 70s to 90 range. And that additional 10% or 30 to 10% is going to be these fatty acids that are basically not coming along in our extraction. They're getting caught in our method. And now we subtract out what we know is present from our beeswax standard. So if it's 1 gram of beeswax and we end up with 0.7 grams, obviously if we use 10 grams RLD, we know the RLD is 8% beeswax, we can subtract that out. So now everything that's left, at least in our calculation, is mineral oil and petrolatum. So we have a total mineral oil and petrolatum number. That's where we get the 89% from. Now the challenging part here is, what's the ratio? Well, how we do that, like you saw on the last slide, is we run a series of standards. Those standards will be in different ratios, maybe 90-10 all the way down to 10-90 of mineral oil to petrolatum ratio. We identify the closest matching mixture of standards, and now we're going to prepare some closer ones and find that ratio that works. So you'll see on the top left here, a mixture that's 58-42, where you're going to compare it to the RLD. We may even add some of that mixture to the RLD to see how it changes. Does it change the way we expect? Does it look very similar? And that's kind of how we're going to give you the grade and the ratio. So of that 89%, in this case, you would see 58% of the super white protopet, which is a white petrolatum, 42% of the Dracol 35. So that's kind of the overall process of how we handle a really difficult project like these mineral oil petrolatum, even beeswax added in, white wax, paraffin wax, things like that. How do we handle those? This is a really good representation of how we have a ton of experience doing this. And quite frankly, you're not going to find anybody better to do this type of testing with the methodologies we've developed. So let's go into the next formulation, for example, which is our solid oral. This is a tablet formulation I will note that everything that I've gone through in this presentation is made up in a way. We're not showing any of our client data. So what you'll see here is a formulation that contains lactose, stearate, magnesium stearate, microcrystalline cellulose, colloidal silicon, and carboxymethylcellulose sodium. So why is this formulation so difficult? It's those three in the middle, the microcrystalline cellulose, colloidal silicon, carboxymethylcellulose sodium. And the reason is they're not soluble. We're not going to be able to separate them through solvents. We're not going to be able to separate them through any of our different testing techniques. So how do we attack a project like this? Well, first and foremost, lactose can be done by HPLC. Magnesium stearate can be done through total magnesium, like an ICP measurement. Stearate can be done through a FAME prep. In looking at the stearic acid FAME prep, we can get a stearate number. We compare them. We're done with the first two. Lactose and magnesium stearate are done. So let's talk about how we approach the last three. So when we talk about these, one thing to note is magnesium, when we do this, is going to come with us. So what we're going to do is a targeted solvent extraction preparation. We're going to do preparations to remove the lactose, remove the API, remove the magnesium stearate. Now, when we remove the magnesium stearate, the magnesium is not soluble. It's not going anywhere. It's going to remain behind. So we isolate those insolubles. We're going to do an ICP OES for silicon, sodium, and magnesium. Now, the nice thing here is colloidal silicon, if we run a standard alongside, we can quantify silicon in the RLD, quantify silicon in the standard, and now we know how much silicon is attributable to colloidal silicon in the RLD, and we can back that one out. So finally, we're just left with microcrystalline cellulose and carboxymethylcellulose sodium. So how do we tackle that? Well, again, we're going to run standards alongside through the whole extraction procedure. We're going to run standards when we run the ICP OES. We're going to use a sodium standard, a carboxymethylcellulose sodium standard, to quantify total sodium. Now, this is an approach that we've developed over time. It's a very difficult process for the simple fact that we need to extract the lactose out with water. Well, a lot of the times these celluloses will expand, they will swell, they will float in water, which makes extracting them very difficult, because when you think of an extraction, what you're doing is adding solvent in, you're vortexing it, you're centrifuging it down, you should have a solid pellet at the bottom and clear liquid up top. That doesn't always happen here, and it takes a lot of work to do this. So at the end of this, what we're hoping to do is using these ICP values as well as other techniques we may have available, like FTIR and Raman, to kind of help us distinguish these materials. We're going to use stoichiometric calculations to speciate out silicon, sodium, and also this microcrystalline cellulose. And at the end of the day, we want to report with these plus minus values, which are associated with our RSDs and RPDs. So this is a really challenging project. I need to stress that. These materials are very difficult to separate from each other. And at the end of the day, we figured out methods that we are very confident in doing this with. So I have a lot of confidence in our team to be able to do this, and you should too. So finally, we're going to get into an example ophthalmics. And these are some of the most fun formulations, and not just because they're 99% water and we're almost done. They're really fun because when we look at these formulations, there are so many moving parts and so many things that just everything seems to have a counter ion that matches something else. So it's fun because it's tough. And a couple things that we need to go through here. So obviously we have a buffer system. We have these other materials that have chlorides and sodiums. We've got hydrochloric acid and sodium hydroxide listed on the insert. So how are we going to do it? Well, first thing, hydrochloric acid and sodium hydroxide are considered non-critical excipients. So what does that mean? That means they're only added in for pH adjustment, which means we don't have to really report them when we talk about submitting a Q1, Q2. So we've eliminated that. We're not going to worry about hydrochloric acid. We're not going to worry about sodium hydroxide. So how do we do this? How do we go about this approach? Well, like I said, everything seems to have a counter ion. Well, the good news is not everything actually does. There are things in here that are unique that we can start to, by process of elimination, get to our answer. So how do we get to that answer? Well, the answer is total sodium, total phosphate, total chloride. It's going to come from all sorts of sources, but we're going to get a total number. We're also going to do editate sodium, or EDTA, by HPLC. We have really good and well-established methods. Basically, it's an iron chelation method. We're going to quantify EDTA. We're going to associate a certain amount of sodium with that and pull that from the total. And now our total number is less because we pulled some towards the EDTA because we know how much sodium is associated with that. The same is true for benzalkonium chloride. We're going to do that by LCMS. We're going to get a quantitation. We're also going to run a standard of benzalkonium chloride when we do our ion chromatography. And we're going to subtract out the chloride we can quantitate from that standard from our total chloride. What's left is our total chloride, and we've done benzalkonium chloride. So we're done now. When we talk about our next steps, so now we still have sodium chloride, we still have sodium phosphate, monobasic and dibasic that we need to quantify. How do we get there? Well, when it comes to sodium chloride, it's pretty simple now. The remaining chloride we associate with sodium chloride. That's how we get to that point. Once we're done with benzalkonium chloride, we associate the rest of the chloride with sodium chloride. That pulls another piece of sodium away from the total. So now what we're left with is a total sodium, total phosphate. Everything left should be associable with sodium phosphate, dibasic and monobasic. Now we're going to go back to Gen Chem, and we're going to use the Henderson-Hasselbalch equation, which is great for buffer systems. We know the pH because we're going to measure that of the as received material. And the ratio at which you add sodium phosphate, dibasic and monobasic, in theory, should be in line with the pH. And using that Henderson-Hasselbalch, we're able to calculate the total sodium dibasic and monobasic that we have. Now, the nice thing to know here is that the sodium and phosphate numbers that we have left over after all these calculations should add up. We should have these numbers add up to very similar. Sometimes they're a little bit off. Again, every measurement has an error. But at the end of the day, this is some methodology that we've worked on developing over the years. Again, I'm really proud of this. I worked on this in the lab, and it's something that we have found great success with. One thing you'll notice I didn't mention is the water in the API. So the water, again, Carl Fisher, it's going to be 99%. Sometimes we don't even have to measure it because we know it's going to be the rest of the formulation, right? So we do every other measurement. Maybe we get 0.8%. We know 99.2 is the water. We sometimes do it. We sometimes don't. Really just depends on what we're looking to do. The API in most situations might also have a counterion. So like the example with benzalkonium chloride, part of the chloride that we see can come from these benzalkonium chloride species. But the API may have chloride, the API may have sodium, and we might have to do the exact same thing with the API in this whole mess that we have here. The good news is we can do that. So again, we've developed these methodologies, we're really confident in them and how they work, and we've seen a lot of really great success with them. So at the end of the day, with these ophthalmics, with these solid orals, with these topicals, I showed 3 pretty difficult examples. We've seen a lot easier formulations than these and had great success. These are some of the more difficult ones in terms of the procedures that we have to follow. And again, we've shown that these methodologies work time and time again, and we're really confident in them. So why work with EAG, urethans in general? So first of all, client confidentiality is really important to us. We have a lot of support systems. We have a lot of moving parts that go towards making sure that your data is safe with us and your methodology is safe with us. Also, we have some of the best people in the world when it comes to these types of deformulation activities. You're not going to find a better lab. EAG is the best when it comes to this testing, this deformulation testing. Dr. Uffman, who is going to be helping with the Q&A, actually is one of the pioneers of doing this testing. We have a ton of experience. Again, we have some of the best people in the world to do this. But not only that, we have so much support from our parent company. We also have a ton of testing capabilities beyond what we've talked about today. We offer the ability to move faster. If you need to rush your deform, if you have a deadline, we can help you hit that. We have a ton of instrumentation too. We have redundancies in instrumentation, which means those timelines that we talked about, we're hitting most of the time, unless we run into issues that we don't expect. We are the global leader in this type of testing and other types of testing. When it comes to deformulation, though, you're not going to find a better lab to work with. And we're also ISO 9001 and 17025 accredited. 9001 is a really important accreditation for us. It's a critical thinking accreditation. That's what we do. We think to solve your problem. That's why we got into deformulation. They're not straightforward. They are tough tests, and we have found ways to make it work. We're really excited to share that with you. So I want to thank everybody for joining the webinar today, and I'm going to hand it back over to Melissa to start the Q&A.